The overall goal of this procedure is to demonstrate an optimized protocol for successful visualization of sound perception and processing in auditory and song control regions in the songbird brain using functional MRI. This is accomplished by first preparing good quality auditory stimuli, which can be incorporated into an on-off block paradigm to present to the bird during functional MRI or fMRI scanning. The second step is to prepare the bird for scanning by first anesthetizing it and then by positioning it on an adapted animal bed, which is subsequently placed in the bore of the MRI scanner. Next, fMRI images can be acquired during auditory stimulation. The final step is to pre-process the acquired MRI data, including normalization of the images to the zebra-finch atlas. Ultimately, statistical voxel-based analysis is used to allow visualization of the obtained results and localize the brain activations induced by the auditory stimuli.
The main advantage of this technique over other more commonly used methods in songbird research, like electrophysiology, histology, and molecular mapping techniques, is that fMRI is a non-invasive in vivo method which allows one to study the same subject over time and to address the whole brain at once without prior hypothesis on the localization of the neural substrates involved in the investigated process.
Individuals new to this method may struggle because implementing functional MRI in birds is very different and far more challenging than using the same method in humans or rodents. This is not only due to the small size of their brains of these subjects, but also because their skull comprises of numerous air cavities which induces susceptibility artifacts. Now, by using a spin echo sequence, a sequence less susceptible to these artifacts compared to a gradient echo sequence, we can do whole brain functional imaging of the songbird.